The overall goal of this procedure is to assess ploidy of metaphase II eggs after experimental manipulation. Begin by collecting fully grown mouse oocytes from primed sexually mature females. Then microinject the oocytes with materials of choice to perturb the protein of interest, and mature oocytes in vitro to metaphase of meiosis II. Next, stimulate chromosome spreading in the egg by treatment with monastrol, which results in a monopolar spindle. Ultimately, these results can depict ploidy status of the egg through immunofluorescence and confocal microscopy.
This method can help answer key questions in the reproductive biology and chromosome segregation fields, such as understanding the molecular mechanisms that lead to aneuploidy.
Visual demonstration of this method is critical, as the microinjection technique is difficult to learn because several steps are hard to convey in writing.
Intraperitoneally inject sexually mature female mice with 5 IUs of pregnant mare's serum gonadotropin to maximize the number of antral follicles isolated from each mouse. Approximately 48 hours after PMSG priming, warm 3 ml per mouse of collection medium at 37 degrees Celsius and equilibrate 1 ml of culture medium for at least 1 hour. Set up microdrops of each in a petri dish and overlay with mineral oil. Now place the culture medium dish into the incubator and the collection medium dish on a slide warmer. Sacrifice the mouse, dissect the ovaries, and place them into a watch glass containing pre-warmed collection medium. Using a 1 ml insulin syringe, anchor the ovaries to the dish and release the antral follicles by puncturing them several times with 27-gauge needles that are fastened together. Under a dissection microscope, collect cumulus oocyte complexes using a mouth-operated glass pipette. Only collect large antral follicles and not the smaller pre-antral follicles or denuded oocytes. Transfer the complexes to a microdrop of prepared collection medium. With a small pipette slightly larger than the diameter of the oocyte, detach the cumulus cells from the complexes. Transfer the denuded oocytes with a larger pipette into a microdrop of culture medium. Allow oocytes to recover for at least 1 hour in the incubator prior to manipulation. Make injection pipettes by pulling borosilicate glass capillary tubing in a mechanical puller. Remove the media chamber from a 1-well chamber slide. To create a microinjection platform, place a 5 microliter drop of collection medium as close as possible to a 0.5 microliter drop of siRNA, cRNA, or morpholino oligonucleotide injection material. Cover with mineral oil and place on the microscope stage. Place injection and holding pipettes into the holders and position into the drop of collection medium. Turn on the nitrogen tanks that are connected to the pico-injector and anti-vibration table. Open the tip of the injection pipette by gently tapping it against the holding pipette while filling it with medium. Transfer 5 to 10 oocytes from the incubator to the collection medium drop on the platform. Now, set the parameters of the pico-injector for an injection volume of 5 to 10 picoliters. While pressing clear, move the injection needle to the material drop and fill. Return it to the medium drop. Use the holding pipette to capture an oocyte and align the injection needle, oocyte, and holding pipette along the x-axis. Under higher magnification, advance the injection pipette through the oocyte, being careful to avoid the nucleus. Once the plasma membrane is pierced, press inject. After injection, withdraw the needle. Release the oocyte and repeat. Once all the oocytes are injected, return them to the incubator to hold in culture medium for the desired time, which typically ranges from 12 to 24 hours. Then wash the oocytes through several drops of CZB and transfer to a microdrop of CZB under oil and incubate for 16 hours to complete maturation to metaphase of meiosis II. Place 750 microliters of CZB containing 100 micromolar monastrol into the well of an organ culture dish that has water in the outer ring. Now wash the eggs through several drops of CZB plus monastrol and then transfer them into the organ culture dish and incubate for an hour. Fix the eggs in a watch glass containing 2% paraformaldehyde and PBS for 20 minutes at room temperature. Next, transfer eggs into blocking solution and store at 4 degrees Celsius until processing. Permeabilize eggs for 15 minutes at room temperature, then rinse through several large volumes of blocking solution. Perform the rest of the procedure on the lid of a 96-well plate that is protected from light in a humidified chamber at room temperature. Transfer eggs to 25 microliter drops of blocking solution for 15 minutes. Then, to label centromeres, transfer eggs to a drop of blocking solution containing Crest antiserum at a 1:40 dilution. Incubate for at least 1 hour, then wash through 3 drops of blocking solution, incubating 15 minutes in each. Transfer eggs to a drop of blocking solution containing fluorophore conjugated antibody and incubate for 1 hour. Wash twice in blocking solution. To detect chromosomes, incubate in a 1 to 5,000 dilution of Cytox green in blocking solution. Mount samples in 5 microliters of VectaShield. Put 4 small spots of petroleum jelly at the intended position of cover slip corners to prevent crushing of the eggs. Place the cover slip and seal with nail polish. Store in a slide box at 4 degrees Celsius until processing via confocal microscopy. While imaging with a 100x objective, capture 0.4 micrometer steps in the Z-plane and image the entire region of the metaphase spindle. Count centromeres using imaging software such as ImageJ from NIH. Euploid mouse eggs contain 20 kinetochore pairs, giving 40 total spots. In this Z projection of a euploid metaphase 2 egg, DNA is colored in green and kinetochores are colored in red. The arrows point to 2 distinct chromatid arms, indicating overlap at kinetochores number 17 and number 18.
Following the microinjection procedure, other methods like Western blotting, real-time PCR, spindle configuration, and chromosome alignment assessments, and in vitro fertilization can be performed in order to answer additional questions like how perturbation of your protein of interest affects meiotic maturation and developmental potential.
After watching this video, you should have a good understanding of how to manipulate oocytes and assess ploidy of metaphase 2 eggs.